In this lecture, I am going to explain the principle, design and construction of bioreactors. Before that, we should note what is bioreactor. You all know it is nothing but any of the vessel that is used for the growth of microorganism. It is otherwise called as fermenter. The major role of fermenter is in producing aseptic condition for the fermentation. So during fermentation, the bioreactor not permit any contamination. The exact definition for bioreactor is an apparatus used for growing organisms such as bacteria or yeast that are used in the biotechnological manufactures of many substances like pharmaceutical products, antibiotics, vaccine, and for the bioconversion of many organic wastes. Next, we should know about fermentation. It is nothing but a metabolic process that converts sugar materials into either acids or gases or any alcohol. We all are know and most of the fermentations are carried out by many bacteria and yeast, and it is used for the production of a variety of chemical products. Let us see the basic principles of bioreactor design. Before that, we should know what is the major function of the bioreactor. I already told bioreactor is provide a suitable environment for the growth of microorganism and can efficiently produce a target product. This is the major role of bioreactor. So, with the help of bioreactor, we can produce a large amount of cell biomass, cellular metabolites, and it is used for the conversion of many organic wastes into beneficial product. The performance of any bioreactor depends on the following key factor. Agitation rate, it is nothing but how the contents inside the bioreactors are mixed. Next one is oxygen transfer. Third one is pH, temperature, and fifth one is foam production. So these are the parameters which are mainly influence the performance of any bioreactor. Then, while designing a bioreactor, we should consider the following factor. Number one, the production organism. That means what organism should be used in the bioreactor. It is a bacteria or fungi or any other organism. Depends upon the organism type, we can design a bioreactor. Second thing, the optimal operating condition required for target product formation. The operation condition is differ from one product to another product. So we should aware about the operation condition required for the particular product. The third thing is product value, whether our product is highly value or moderately value or low value. Fourth one, the scale of production. It is nothing but how much product we need, whether large amount or low amount of products to be produced. And the last one is capital investment, how much amount of cost took for production of one product. Next, we should know about the requirements of bioreactor. A good bioreactor needs the following things. Number one, the design and construction of bioreactor. It must keep sterility from the start point and to the end point. So, the sterility should be maintained from the starting point to end point. Then, optical mixing with low and uniform shear, adequate mass transfer of oxygen, clearly defined flow conditions, feeding substrate with prevention under or overdosing, suspension of solid material, gentle transfer of heat, ability to sterilize for long term, simple construction, simple measuring and control, scale up and flexibility, and the last one is compatibility with low downstream process. These are the parameters should be required for any good bioreactor. Next, while designing a bioreactor, some basic points should be considered. These are productivity and yield of product. Second one, fermenter operation and reliability, product purification, water management, energy requirements, waste treatment, and operation should be contamination free. So these are the parameters we should consider before designing a bioreactor. In older days, the fermentations were carried out in open cylindrical or rectangular vessels. It is made by wood or stone. But nowadays we are using glass material or stainless steel for the construction of bioreactor. A good bioreactor should have the following things. Number one, heat and oxygen transfer configuration. Number two, sterilization procedure. Number three, fast and thorough cleaning system. Number four, proper monitoring and control system. While designing a bioreactor, the following parts or parameters should be included. They are a motor connected with a central shaft and a shaft connected with impeller, acid or base gauge, pressure gauge, an outlet for waste gases, cold water outlet, pH probe, temperature probe, sparger. It is used for proper oxygen supply, steam pipe, harvest pipe, cold water inlet pipe, cooling jacket. It is filled with cold water, oxygen concentration probe, inlet pipe for nutrient or inoculant, steam and antifoam agent. These are the parts or components of a good bioreactor. The construction of bioreactor required steel or glass vessels, which gives a place or support for the growth of microorganisms and their product. I already told in ancient days wooden or rock vessels are used for fermentation process, but nowadays a modern bioreactors are constructed with glass or steel material. Next thing is impeller, which is used for proper mixing of components inside the bioreactor. There are four types of agitator or impeller are used. So with the help of mixing, we can achieve a bulk fluid and gas phase mixing, air dispersion, oxygen transfer, heat transfer, suspension of solid particle inside the bioreactor, and maintenance of uniform environment throughout the vessels. The third one is baffle. It is another part of bioreactor which is used for agitation. The baffles are made up of metal strips roughly 1/10 of the vessel diameter, and they are attached radially to the wall of bioreactor. In majority of the bioreactor, there are 4 baffles are normally incorporated into agitated vessels in order to prevent a vortex and to improve aeration very efficiently. The fourth one is sparger. It is nothing but the aeration system of bioreactor. Normally, the sparger has a pipe with a minute holes, and the hole allows the air inside the bioreactor. Hence, the medium of the bioreactor gets sufficient air supply, followed by the sufficient air supply may enhance the growth of microorganisms. Sparger is normally made up of sintered glass material or ceramics or any other metals. There are two types of forgers are there. One is orifice forger. Second one is nozzle forger. In a orifice forger, there are many holes or drills on the under surface of the tube, it making many rings or cracks. But in case of nozzle forger, it has single hole. The air is supplied with a single hole of sparger in a center of bioreactor. Next, we will see the control and monitoring probes such as temperature probe, pH probe, foam probe, cooling jacket, heating coil, dissolved oxygen probe, and pressure. Normally, maintaining a required temperature for microbial growth is essential for good yield, and fluctuation in temperature may cause damages to microorganisms. So, we should monitor the temperature throughout the fermentation process. The changing of temperature inside the bioreactor may affect solubility and diffusion of oxygen in the fermentation broth. That's why we can monitor the temperature very carefully. Next, pH. The changing of pH of the medium may also affect the growth of microorganism and also cause changes in the yield. That's why we should monitor the pH also. Next, foam checking probe. In many fermentation process, the formation of foam is very undesirable phenomenon. It causes a loss of an essential part of fermentation broth. So the monitoring of foam formation is very essential in the bioreactor. After forming the foam, it can be eliminated by adding anti-foam agents. When foam is rises in the bioreactor and touches the probe tip, then the pump is activated and then anti-foam agent is released into the bioreactor. There are 4 mechanical anti-foam devices are used. Number 1, disc. Number 2, propeller. Number 3, brushes. And number 4, hollow cones. These mechanical anti-foam devices are attached to agitator shaft above the broth surface. The role of mechanical anti-foam devices are, while the foam is formed inside the bioreactor, it breaks the foams. Next, cooling jacket. It is filled with cool water. Normally, the activity of microorganism inside the bioreactor may produce some heat. The heat may affect the process of fermentation and also it denatures some enzymes which are involved in the fermentation process. That's why we should prevent the rising of heat in the fermentation process. The cooling jacket circulates the cooling water in order to remove heat produced by the microbial activity. After that, the fermentation reaction will come to optimal condition. Next, Heating coil, I already told, during microbial activity the heat will be produced. In some of the cases, the fermentation process needs some more heat for getting their optimal condition and also produce byproducts. So, in order to raise the temperature of fermenter, the fermenter is placed in a thermostatically controlled water bath. The water bath has internal heating coil that circulate the hot water around the tank. Then dissolved oxygen probe. It is made up of stainless steel. The dissolved oxygen probe indicated the oxygen level of the fermentation medium. Suppose the oxygen level is get low in the fermentation medium, and it is managed by increasing the speed of agitator in the fermenter. And the last one is pressure gauge. The most of industrial bioreactors are designed to withstand a specific working pressure. The measurement of pressure inside the bioreactor is one of the major important safety measures. So we should monitor the pressure very carefully during the fermentation process. The measuring of pressure can be achieved by with the help of pressure gauge. There are three types of pressure gauge are used. Number 1, Bowden tube pressure gauge. Number 2, diaphragm gauge. Number 3, piezoelectric transducer. So, while designing a bioreactor, all of these parameters should be considered. The factors such as vessel shape, agitation, aeration, and baffles, which are play a major role in productivity. So, there are other novel approaches are developed in order to increase the productivity of sparger, agitator, as well as the various controlling devices in order to improve the productivity of bioreactors. Next, we are going to study about the classification of bioreactors based on various parameters, different types of bioreactor and its working mechanism, also their advantages. Now, let us see the classification of bioreactors. Bioreactors are classified into many types based on the 5 following parameters, such as the presence or absence of oxygen, also the requirement of stirring, based on the mode of operation, based on the method of growing microorganism inside the bioreactor, based on the microbial agent which is used in the bioreactor, and based on the process requirement. Let us see the first parameter, the conventional classification based on the presence or absence of oxygen, also the requirement of stirring. So in this classification, the bioreactors are classified into 3 types. Number 1, non-stirrer and non-aerated bioreactor. In this type of reactor, there is no need of stirring, also aeration. That means no mixing and no air supply is involved. These type of reactor are commonly used for the production of some traditional products such as wine, beer, cheese and many beverages. The second one, non-stirrer and aerated reactor. In this type of reactor, aeration is supplied in the reactor but no mixing is applied. This type of reactor are rarely used bioreactor. The third one is stirrer and aerated reactor. In this type of reactor, both stirrer and aeration is applied. This type of reactors are commonly used for the production of many microbial metabolites such as organic acid, vitamins and many antibiotics. The second parameter, based on the mode of operation, the bioreactors can be classified into 3 types. Number 1, batch reactor. Number 2, fed batch reactor. Number 3, continuous reactor. The batch reactor is very simple one. In this type of reactor, we will supply the nutrient at one time and harvest the product at one time. This is the batch fermentation. But in case of fed batch reactor, we are supplying the nutrient at one or more time but harvesting the product at one time. This is the difference between batch reactor and fed batch reactor. In continuous reactor, the nutrients will be supplied at many time and also the product is harvested at many time. These are the major 3 classification of bioreactor based on the mode of operation. The third parameter, based on the method of growing microorganism inside the bioreactor. Based on this parameter, the bioreactors are classified into 2 types. One is suspended microbial growth. Second one is immobilized microbial growth. In the first type, free microbial cells are directly suspended into the bioreactor But in case of second type, that means immobilized microbial growth, the microorganisms are immobilized with some other particles. The immobilized microbial growth reactors are further classified into four types: moving bed bioreactor, fibrous bed bioreactor, packed bed bioreactor, and membrane bioreactor. These are the four sub-classification of immobilized microbial growth bioreactor. The fourth parameter, on the basis of the microbial agent used. In this type, the bioreactors are classified into two groups. One is living microbial cell, second one is employing microbial enzyme. In the first type, living microbial cell, the fermenter is containing the living free microbial cell for the fermentation. But in case of second type, employing microbial enzyme, instead of microbial cells used here, the microbial product, that means some enzymes are used for fermentation. The last parameter based on the process requirement, that means based on oxygen supply and the state of reaction, the bioreactor are classified into four groups. Number 1, aerobic bioreactor. Number 2, anaerobic bioreactor. Number 3, solid state bioreactor. And number 4, immobilized bioreactor. In the aerobic bioreactor, the reaction is carried out in the presence of oxygen. In case of anaerobic bioreactor, the reaction is carried out in the absence of oxygen and the presence of carbon dioxide. So in the third type, solid state, the name is enough. The reaction is performed in solid state instead of liquid medium. The fourth one, immobilized type. In this type, the microorganism involved in bioreactor immobilized with some other particle. These are the common classification of bioreactor based on many parameters. Next, based on various parameter and classification, finally the bioreactors are divided into 7 types. They are batch reactor, continuous stirred tank reactor, air-lift bioreactor, fluidized bed bioreactor, packed bed bioreactor, photobioreactor, and membrane bioreactor. So these are the 7 major types of bioreactors. First, let us see the batch bioreactor. In this type of reactor, the medium, that means the cultural medium, is added at one time, and also the product is harvested at one time. There is no addition or removal of medium will take place. The process will complete at one batch. That's why the reaction is called as batch reaction. The batch reactor is mainly operated with mixing. That means this type of reactor are fitted with many propeller. It helps to remove the concentration gradient and also provide the homogeneous environment to the entire bioreactor. So the components in the reactor are distributed uniformly throughout the reactor. In this type, the bacterial requirement such as electron acceptor and nutrients are sufficiently high in concentration, and that they are impose no limitation on organism and growth rate. These batch bioreactors are commonly used in the production of many microbial metabolites in industry as well as in the research laboratory, and also many pharmaceutical products. The advantages of batch reactor are flexibility. We can easily perform the batch reactor, and also it is easy to operate and relatively low cost of equipment and also maintenance. The major remarkable feature of batch reactor is the possibility of using directly to the research laboratory. These are the major advantages of this batch type reactor. Next, continuous stirred tank bioreactor. In this type, the contents of the vessel no longer vary with time, and this applies to the hold up microorganism and the concentration to the components of the medium in the fermenter. In this type of reactor, the medium is supplied at many times, and also the product is harvested at many times with continuous agitation. This is the important thing. In continuous stirred tank reactor, commonly yeast and bacteria are used for the production of the primary metabolites such as enzymes and amino acid, and also the production of energy. It is the best example is the production of alcohol. Also, this type of reactor is commonly used in activated sludge process, which is employed in wastewater treatment. The major advantages of continuous stirred tank reactor are it is continuous operation. We can operate this type of reactor continuously with good temperature control, and we can easily adopt 2 phases of run, then simplicity of construction and also low operating cost, and finally it is easy to clean. The third type is airlift bioreactor. This type of reactor, it is working on the principle of an airlift pump. Based on airlift pump, it is further classified into two types, internal loop type and external loop type. In this type of reactor, the reactor's volume is determined by its capacity, kinetic data and specific growth rate of the microorganism, and the rate of airflow of the reactor is depends on the volumetric mass transfer of coefficient in the reaction system. The advantages of airlift bioreactor are simple design with no movable parts. It can easier to sterilization. We can operate this type of reactor with low energy. In this type of reactor, heat produced during the process is easily removed and also very low cost. Next, fluidized bed bioreactor. In this type, the fermenter is filled with solid particles which are suspended in an upflowing liquid stream, which is supplied at the bottom of bioreactor. The solid particles present in the bioreactors are easily distributed throughout the bioreactor, so that the smaller particles are found at the top of the fermenter and also the higher particles are found at the bottom of the fermenter. This type of reactor are commonly used for the production of alcohol with flocculant yeast. The advantages are uniform particle mixing, uniform temperature gradient, and ability to operate this type of reactor in continuous state. Next one, photobioreactor. This type of reactor are generally made with glass tube, and the major thing is the fermentation is carried out by either exposing to sunlight or any artificial illumination. That means the reaction is only carried out in the presence of light. This photobioreactor is commonly used for the cultivation of many algae for the production of keratin, xanthine and many metabolites from algae. The advantages are higher productivity, large surface to volume ratio. That means we can cultivate the algae at larger area, better control of gas transfer, reduction in evaporation of growth medium, more uniform temperature, better production from outside contamination, and also space saving. The last type is membrane bioreactor. In membrane bioreactor, the soluble enzyme and substrate are introduced on either one side of ultrafilter membrane by means of pumping. And with the help of this pump, the product is forced out throughout the membrane. The contents of the reactor is mixed properly with the help of stirrer. The commonly used membrane material in membrane bioreactor are polysulfonate, polyamide and cellulose acetate. The advantages are the loss of enzyme is reduced in membrane bioreactor compared to other type of reactors, and also the major thing is we can easily replace the substrate and enzyme. Okay, these are the major types and application of various bioreactor. Thank you.